Hello, I'm Chris Goldring. I'm a professor here in Liverpool. I've been in Liverpool 26 years in this department. I am co-lead for the Human Liver Research Facility within Koppel. I am also Deputy Dean of my institute, Institute of Systems, Molecular and Integrative Biology. The Human Liver Research Facility is something that has grown out of the need to isolate human samples and to derive human-based models. What really gets me enthused about the work that we do is the fact that we can look at mechanisms of adverse drug reactions as they actually occur with real, true human patient samples. So these are samples which have been acquired sometimes only 30 minutes previously in the hospital next door here in Liverpool. It allows us to be able to understand what is actually happening in this key tissue, which unfortunately so often gets affected adversely by medicines where there is an adverse drug reaction problem. And it allows us to dig into the detail of those adverse mechanisms to allow us to translate that into real findings to help to make new medicines safer.
Hi everybody, my name is Dr. Giusy Russomanno and I'm a postdoctoral research associate at the Department of Pharmacology and Therapeutics at the University of Liverpool. I'm currently working on developing a 3D spheroid model made from human liver cells, which basically resemble mini livers from patients without the use of any animal-derived material, to study how medicines can affect patients with fatty liver disease. This condition, which is characterized by the accumulation of fat within the liver, affects about 1 in 4 people worldwide, and it often goes unnoticed because patients do not have any symptom. However, despite not being a life-threatening condition, patients with fatty liver disease might have more or more severe side effects when taking certain medicines. Current research in this field sadly relies heavily on the use of animal models, such as mice, rats, but also guinea pigs and hamsters, which do not reflect at all human biology. So it is very important in this moment that we actually look for animal-free alternative and more human-relevant models that can better translate the research finding into the clinic and ultimately make medicines better for those patients.
This is really important that we evolve and we change and we also revolutionize the way that we do our work, avoiding where we can the use of animals in research, because of the fact that those animals, any data that you generate using an animal in research, will not actually help to successfully generate a safe and effective medicine that can be used in the clinic. So essentially, many of those animal experiments are just not fit for purpose, and they just won't be useful in generating a new medicine. That is aside from the obvious ethical issue of using or overusing animals in research as well.
So in research, we need to use enzymes to break down tissue into individual cells that can be grown in the lab. Traditionally, this has always been done using an enzyme called collagenase, which was originally isolated from rat tail. But also its new, more modern form, generated using bacteria, still contains quite a lot of significant mammalian components. For this work, we decided to switch to Liberase, which is a high-purity enzyme that does not contain any mammalian raw material. This does not only means that you have less risk of contamination in your experiments, but also it increases consistency and reliability. One thing that I feel is really important is that thanks to Animal Free Research UK funded experiments, we now use liver rays exclusively in our facility for the isolation of all human liver cells. And this does not only have very big impact in terms of use of animal-derived materials within our department, but it also helps us raise awareness across other researchers and prove that animal-free alternatives are available, and sometimes they can be even better than what is considered to be the gold standard.